Our next speaker today is Dr. Montserrat Anguera. She got her undergraduate degree from UC San Diego, where she studied environmental chemistry. And she did her PhD with Dr. Patrick Stover at Cornell. And there she worked on folate metabolism using cell culture and mouse models. Because of her interest in epigenetics, she joined Dr. Jenny Lee's lab at Massachusetts General for her postdoc, where she worked on long non-coding RNAs in a range of systems, including the X-chromosome in mouse and pluripotent stem cells. In her own lab, now at UPenn, where she recently got tenure, Montserrat studies X-chromosome inactivation and how this process contributes to female-biased autoimmunity. I first heard Montserrat speak at the Cold Spring Harbor Epigenetics meeting a few years ago, and not only was her talk fantastic, but throughout the span of the meeting, Montserrat's trainees could not stop raving about her excellent mentorship. So without further ado, it is an absolute pleasure to have you in our seminar series, and we are very excited to hear about your talk.
Okay, thank you so much. And that was such an amazing introduction. What I want to tell you guys about is a story about finding something that is right in front of you. And if you don't look, if you don't ask the questions, if you don't do the experiment, you'll never know, right? So this is a picture here of Machu Picchu. I was super lucky to be able to go there last year, and it's a story like of Machu Picchu, right? This beautiful, these ruins have been there for such a long time, and it wasn't until very recent that they were discovered. So what I'm going to tell you about today is about X-inactivation. And in my lab, we're studying how X-inactivation is maintained in the immune system. And the reason why it was such a sort of crazy discovery is that it was traditionally thought that X-inactivation happens in the same way no matter what type of somatic cell that you are. And what would make an immune cell so different, right? And so that's what I want to share with you today. So first, I just want to show you some of the data that's been known for quite some time from the clinical field, and that is that there is a very strong sex bias when it comes to some autoimmune diseases. So there's about 80 different autoimmune disorders, and 25 of those have a very strong female bias. And that's shown in this graph right here. So the name of the disease is shown on the left, and the pink bars indicate the percentage of patients that are women compared to men. So you can see sort of the top 5 autoimmune diseases that are biased towards women include Hashimoto's, lupus right here is number 5. So it's about 85% female. And so what's important to note is that this sex bias has been described in the clinical literature for a long, long time. Yet what's been missing is a molecular understanding of why having 2 X chromosomes predisposes an individual to get autoimmune disorder. And so a variety of hypotheses have been proposed over the years, but none of these has been rigorously tested. And so for some reason, having extra copies of an X chromosome increases your risk to develop lupus disease and also Sjogren syndrome. And that's shown right here. So if you have just 1 copy of an X chromosome, such as men, right, XY, or individuals with Turner syndrome, they're at very low risk for developing these autoimmune diseases. So then one could ask, well, why is it that having extra copies of an X, such as individuals with triple X syndrome, what's so special about the X chromosome? Well, the X chromosome in the mouse and also in the humans has accumulated a number of very important immunity regulatory genes. And so some of these important genes are shown here. So there's TLR7, which I'll tell you about in the next slide, FOXP3, which is really important for Treg cells, BTK and CD40 ligand. And so evolutionarily, these immune genes have accumulated on the X chromosome. And if you look across the genome, the X has the highest enrichment or density for immunity-related genes across all of the chromosomes. So if you have alterations in the dosage of some X-linked genes, this can be associated with autoimmunity and or result in the development of autoimmune disease. And so I'm just going to share with you 2 examples today. And the first example has to do with this X-linked gene called TLR7. So there was this mouse, there's this mouse that's called the BXSBYAA. And in this mouse, it's a spontaneous mouse model of autoimmunity. In these animals, the males, so 100% of the males develop autoimmune disease. Back in 2006, it was discovered that the reason for this was that there was a change in the dosage of TLR7. So there was a translocation event of a region of the X chromosome that made it onto the Y chromosome. And this region contained TLR7. Thus, all the mice had 2 copies of TLR7, and these animals would develop lupus-like disease, so autoantibodies, and eventually glomerulonephritis, so kidney disease. And what's interesting is that if you make transgenic animals with increased copy numbers of TLR7, they get autoimmunity. If you knock down TLR7 in this mouse strain, you completely eliminate the autoimmune phenotypes. So the second example of an X-linked gene whose dosage is important is CD40 ligand. And in this experiment, Tcels were taken from both male and female lupus patients, and they looked at the expression levels for CD40 ligand. And what they found was that in female patients, there was twice as much CD40 expressed in women, but this difference in expression was not shown or not observed in male patients. And so what the authors suggested was that perhaps the increased expression of CD40 ligand was coming from the inactive X chromosome. Now, there's no direct evidence for this. This is just what was proposed. But I remember reading this, this paper and thinking there's got to be something going on with the inactive X chromosome. And this is something that we can investigate. Okay, so what is X-inactivation? So if you look at the autosomes between the sexes, there's 2 copies of each. And so they're balanced. Their transcriptional output is the same, regardless of whether you're a male or a female. And so if you look at the sex chromosomes in mammals, they're different in numbers, where females have 2 copies of an X chromosome compared to males that have just 1 copy. And so what female mammals have done is evolve this very elegant mechanism for equalizing the expression of genes from the X chromosome, and this is called X-chromosome Inactivation, where they're going to randomly select 1 X chromosome for transcriptional silencing in each and every cell in female mammals. And so X-inactivation is initiated very early during development, and it involves the this expression of a very special gene that encodes for a long non-coding RNA. And this gene is called Xist. So this is the Xist gene right here. And it produces a transcript that is never translated into a protein. It's very long, it's 17 kb. This RNA is spliced and it's polyadenylated, and it's only expressed from the inactive X chromosome. And there's so much of it, and it's so long that you can actually visualize its association along the inactive X chromosome. So in the field of X-inactivation, we use fluorescence in situ hybridization, or FISH, in order to image this long non-coding RNA. And this is a picture of a nucleus of a cell that has 2 active X's that has not yet undergone X-inactivation. So using probes specific for Xist RNA from cells that have not undergone X-inactivation, so this would be found in female embryos in the inner cell mass in the mouse, or in female embryonic stem cells from the mouse. So there's 2 active X chromosomes, and both of them express Xist RNA. And so each of these are the 2 pinpoints, each an active X chromosome. Now, for X-inactivation, either one of these X's will be chosen at random for transcriptional silencing in the process known as X-inactivation. And on the future inactive X chromosome, the Xist gene gets upregulated, and these RNA transcripts remain associated on the inactive X chromosome. So it forms this cytologically visible cloud. And this is the pattern that's observed in differentiated ES cells. It's also observed in somatic cells. And so the dogma of the field is that once X-inactivation has been initiated in the early embryo, this pattern of localization of Xist RNA is maintained no matter what type of cell you are. So after Xist begins to be upregulated, the inactive X chromosome accumulates a number of heterochromatic modifications, and there's so much of these marks that accumulate on the inactive X that we can actually visualize it using immunofluorescence. And that's what's shown in this picture right here. This is sequential RNA fish for Xist RNA. This is the Xist RNA cloud. This is the same nucleus going from left to right. And this blob right here is H3K27me3, which is a silent mark of chromatin. And then this is X paint or DNA fish showing the locations of the 2 X chromosomes. So below here, I've just listed some of the characteristic modifications that become enriched on the inactive X chromosome. So it accumulates DNA methylation, these marks of heterochromatin that are traditionally associated with transcriptional silencing. And so what's important is that once the inactive X chromosome is formed, with each round of cell division, there's a memory of transcriptional silencing. So if that X chromosome was a paternal X, it will be silenced in the daughter cell after cell division. And so this gets maintained into adulthood. And so what we found in the lab is that immune cells are very different from other somatic cells. So it sort of breaks this rule that we had an understanding of, that X-inactivation maintenance is the same no matter what type of cell you are. If you're a neuron, if you're a muscle cell, the immune system works completely differently. And so what I'm showing here are pictures of circulating T cells and B cells from a mouse. It looks identical in humans. And this is RNA fish hybridization. And what you can see is that these cells don't have this characteristic Xist RNA cloud. They're completely devoid of it. Now, these are naive lymphocytes, and when they encounter an antigen in the body, they are activated and they'll re-enter the cell cycle and start dividing like crazy. So then we ask, well, what happens with Xist at this point? And what you can see is that Xist RNA transcripts return back to the inactive X chromosome upon activation. And it doesn't matter if you're a T cell, it doesn't matter if you're a B cell. We see this canonical Xist RNA cloud in these activated lymphocytes. So we wondered, well, does the Xist transcription change in the naive state? And is this why we don't see any localization of Xist RNA to the inactive X? So we looked at the steady state levels for Xist RNA, and we normalized to a total cell number because these are quiescent cells. And what we found is that there is abundant transcription of Xist RNA in the quiescent state, but there isn't localization. And so this is the first evidence of this uncoupling of Xist transcription with its localization to the inactive X chromosome. So Xist is being abundantly transcribed here, but it's not retained at the inactive X chromosome. So next we ask, well, what happens to these marks of heterochromatin that are typically enriched on the inactive X chromosome in somatic cells? So we performed sequential RNA fish, followed by immunofluorescence and DNA fish. So this is the same nucleus going from left to right. And looking at human naive T cells in circulation, what we see is that these marks of heterochromatin are missing. So they're not enriched on the inactive X chromosome in circulating lymphocytes. However, in stimulated cells, in vitro stimulated cells, we see that these marks of heterochromatin, some of them come back to the inactive X, but certainly not all of them. And so what we conclude is that the chromatin of the inactive X chromosome in lymphocytes is different. It's almost more relaxed, more permissive to partial reactivation in these immune cells. So when do these marks actually disappear from the inactive X chromosome? So this is a cartoon showing the lineage of development of lymphocytes. So the lymphocytes are right down here, and their common origin, they originate from HSC, so hematopoietic stem cells right up here. So we wondered, well, if we isolate each of these different cell populations, what happens to Xist RNA and marks of heterochromatin? So we did the following experiment using bone marrow from mice, and this is just a cartoon showing the lineage for development of B cells. And we isolated cells from each of these different stages, And we found that Xist RNA clouds are present in the HSCs and also in the common lymphoid progenitors and the common myeloid progenitors shown right here. Excuse me. However, something really strange happens when the common lymphoid progenitors commit to the pro-B cell stage. Xist RNA completely disappears from the inactive X chromosome. And what's interesting, I don't have time to show you the data today, is that the marks of heterochromatin are still present here. So H3K27me3 forms a focus in these cells, as does H2A-K119 ubiquitinylation. And as these cells undergo differentiation to the immature B cells, these marks of heterochromatin slowly disappear from the inactive X chromosome, and Xist RNA stays off. So what happens to T cells? So T cells originate in the thymus. So we isolated different thymic progenitor cells, and that's shown right here. They're staged depending on different expression of different markers. And just like what happens in the B cells, as the common lymphoid progenitors commit to the DN1 phase of thymocytes, Xist RNA completely disappears from the inactive X. However, unlike in the B cells, in T cells, Xist RNA comes back at the DN2 and the DN3 stage, and then disappears again and stays off through the double positive stage and into the single positive stage. So it has this dynamic movement of coming on and off of the X chromosome. Why has nature evolved the inactive X to be regulated in such a way? We don't know. What happens to gene expression from the inactive X chromosome as T cells differentiate and divide? We don't know. And these are active areas of research that we're looking at right now in the lab. So I just want to also take a moment to share with you that beyond the lymphocytes, other cells in the immune system do very weird stuff when it comes to X-inactivation maintenance. So what I'm showing you here is just the Xist RNA expression patterns in different types of immune cells. So at the top, these are plasmacytoid dendritic cells, and we never detected Xist localizing anywhere within the nucleus in these cells. Xist was expressed, that there are steady state levels of Xist detectable by qRT-PCR, but it never localized. And we activated the cells and we never saw any localization. So this is sort of at one extreme. Looking at the bone marrow for bone marrow derived macrophages, they had small levels of Xist localizing to an area that is the territory of the inactive X chromosome. When they become activated, they get more Xist RNA localizing to the inactive X. NK cells have this sort of dispersed pattern of Xist RNA that doesn't, that extends beyond the territory of an inactive X chromosome. And other types of dendritic cells from the spleen, lymphoid-derived, and also monocyte-derived dendritic cells, they also had these dispersed patterns of Xist, yet other nuclei had canonical-looking Xist RNA clouds. So what does this mean for X-inactivation maintenance? We think that there's very likely to be very different genes that escape X-inactivation depending on whether you're a plasmacytoid dendritic cell versus an NK cell. And this probably allows for very specific and fast upregulation when the animal encounters a pathogen. Okay, so I'm just gonna switch gears now and summarize some mechanistic data in one slide, telling you about how Xist RNA actually gets to the inactive X chromosome when lymphocytes are activated. So in my lab, we've been working on the mechanism, and we found that there are several proteins that are required to bring Xist RNA back to the inactive X in T cells and B cells. And one of these is the transcription factor YY1. And so what we found is that if you delete YY1, Xist RNA becomes mislocalized and it becomes dispersed all across the nucleus, and it looks like this. And we also found that marks of heterochromatin do not return to the inactive X when YY1 is deleted ex vivo. And we also observed that there's female-specific gene expression differences of X-linked genes when you delete YY1 in female B cells compared to male B cells. And so what we propose is that this pattern of Xist mislocalization results in partial reactivation of certain genes on the X chromosome in lymphocytes. And so using that as the basis, as sort of context, I want to share with you some published and also unpublished data looking specifically at lupus and X-inactivation maintenance in T cells and B cells from a mouse model of lupus disease, which is this NZBWF1, and also in lupus patients. So just really briefly, this mouse model of lupus has a very strong female bias. So 100% of the female mice develop lupus-like disease. There's a very strong type 1 interferon response. There's autoantibodies to dsDNA, which is very similar to what's observed in a variety of lupus patients. They also develop immune complexes, which end up in the kidneys. And these animals will die of kidney disease, so glomerulonephritis. So we asked the question, what happens to X-inactivation maintenance as these animals develop disease? So we performed Xist RNA FISH and immunofluorescence analysis looking at H3K27me3 across 3 different stages of disease. So pre-disease animals, early stages of disease, and also late stages of disease. And we classified each of the groups of disease based on the levels of dsDNA autoantibodies in the serum. And so if we look here on the left, what we found is that these NZBWF1 mice, only at late stage disease, have reductions in the percentages of nuclei that had robust canonical Xist patterns of localization to the inactive X chromosome. So what this indicates is that Xist mislocalization was a consequence of the development of lupus-like disease and not a cause of it, because the pre-disease animals looked very similar to wild-type controls. Next, we asked what happens with co-localization of this mark of heterochromatin, H3K27me3. And we looked at the 3 different stages of disease. And what we found was, again, only at late stage disease, there was a reduction in the localization, co-localization of Xist RNA with this mark of heterochromatin. So lupus-like disease affects the maintenance of X-inactivation. So what happens in lupus patients? So we recruited pediatric patients from the Children's Hospital of Philadelphia. These patients are in disease remission. So what this means is that they have their lupus disease, it's well controlled, and they're responding well to medication. So they've been diagnosed with lupus, but they don't have active disease at this point. So we isolated blood from these individuals and healthy age-matched controls, isolated T cells and also B cells, and stimulated the cells in vitro. So what I'm showing here on the left are wild-type healthy controls, and you can see this canonical Xist RNA cloud that localizes. And then on the right is an example of one female lupus patient, and you can notice that the Xist RNA is dispersed all over the nucleus in these cells. So if we quantify the percentages of cells that have the canonical Xist type 1 pattern in healthy controls in lupus, what we see is a dramatic and significant reduction in the numbers of SLE patients that have this Xist RNA localization pattern. So what happens in B cells? Well, we see very similar results. This is healthy controls on the left. These are stimulated for 2 days. And these are the pediatric lupus patients on the right. And when we quantify the percentage of nuclei that have this canonical pattern for Xist localization, we can see that pediatric patients have a very dramatic reduction in the percentages of cells that have Xist localized to the inactive X. So next, we looked at adult patients. So this was a collaboration with the Benaroya Institute, working with Jane Buckner, who has a huge repository of PBMCs from lupus patients with a variety of different disease parameters, active disease, and also disease remission. And so this is healthy samples on the left and an adult lupus patient on the right. And so quantifying the patterns of Xist RNA that have this sort of tight, robust localization at the inactive X, what we found is that lupus patients have, once again, a reduction in the localization of Xist at the inactive X, irrespective of disease status, age, medication. So marks of heterochromatin. So we performed sequential Xist RNA FISH combined with IF in the adult samples, B-cell samples. And what we found was that the marks of this heterochromatic mark H3K27me3 ubiquitin had very dramatic reductions in the co-localization of this focus with Xist RNA in the lupus patients compared to the healthy controls. And so what we conclude is that the chromatin of the inactive X chromosome is affected, negatively affected in lupus-like disease. And so next we ask, well, what happens to X-linked gene expression in lupus patients? And so in order to answer this question, we looked through published data sets of RNA-seq using or looking at SLE patient samples from either Tcels or B-cells. And so I'm going to show you some of the data for both the Tcels and the B-cells. So the study that we got our RNA-seq data sets from is shown below right here. And so we reanalyzed this data set looking specifically at the X chromosome and compared individuals with high SLET scores, so very active disease, compared to healthy individuals. These are all female samples here. And what we found was that 80 X-linked genes were upregulated in lupus. And looking across the X chromosome at the location of these 80 different genes, we mapped where they're located relative to areas that are known to escape X-inactivation. And what we found is that a lot of these areas that are being expressed are in regions that should be subject to silencing, indicating that the integrity of the inactive X chromosome is being compromised in the SLE patient Tcels. So looking at B cells from SLE patients, we also found that there's aberrant expression of X-linked genes, albeit a little fewer than in the Tcels. There are about 53 genes whose expression changes. And the genes that are shown in color right here are either known X-inactivation escape genes, or they're ones that were recently discovered to escape X-inactivation across a variety of B-cell subsets. And what was interesting is that these genes that are overexpressed in the lupus patients compared to the healthy controls are actually expressed at a much higher level compared to the genes that are downregulated. So the chromatin of the inactive X chromosome is really becoming contorted in lupus-like disease, resulting in aberrant expression of genes from the X chromosome. So in this last part, I just want to share with you thinking about mechanisms. So how can these genes become aberrantly silenced? So to kind of get at this, we examined what happens to the gene expression of genes that are important for Xist RNA interactions. So the Xist RNA interactome has been recently identified by, or described in about 5 or 6 different papers using a variety of like genetic screens and also pull down experiments. And so there's about 275 proteins that can interact with Xist RNA. So we looked at this list of 275 Xist RNA binding proteins and looked to see how their expression changes in lupus. So the results for Tcels in SLE are shown here. And these are the genes that are most significantly downregulated in lupus patient Tcels. And what you can see is that there are some genes in here that function in the nuclear matrix, nuclear envelope, chromatin architecture. And what's very exciting for us is that some of these genes have known roles in X-inactivation initiation and also in maintenance for some of them. So in the B cells, there's also a lot more genes that are downregulated that correspond to the Xist RNA interactome compared to genes that are upregulated. And we see this sort of common theme of genes that are misregulated that are important for nuclear architecture, nuclear structure, chromatin regulation, and the list of these is shown right here. Okay, so just to summarize with the model, so what I've told you about today is that in healthy female mammals, T cells and B cells have this dynamic way to maintain X inactivation. So in the quiescent state, Xist RNA and marks of heterochromatin are not cytologically visible at the inactive X chromosome. However, upon activation, when they re-enter the cell cycle, Xist RNA and marks of heterochromatin come back to the inactive X chromosome. And what we see is that there's, I didn't have a chance to share with you, but there's a very specific pattern of genes that are repressed in this state compared to this state. And there's genes that also escape X inactivation. And we propose that the inactive X in the healthy state is maintained in a certain location within the inactive X and also by Xist binding proteins to the matrix around the nuclear periphery. However, in lupus, the situation changes where there's this mislocalization of Xist RNA transcripts across the nucleus. And so there's escape of an additional escape of genes from the X chromosome, causing overexpression of certain genes from the inactive X. And we believe that the Xist RNA interactome, so Xist RNA binding proteins that get downregulated, prevent the regulation of the inactive X chromosome in this disease state. Okay, so with that, I just want to take a moment and acknowledge all the people in my lab that have done all of this work, so both past and present members. A lot of the data that I shared with you today was the work of a very, very talented graduate student, Montserrat, who has now moved on to do a postdoc. This list also includes Ban Pamaru, who worked on a lot of the human B cell stuff, Janla, who was one of the original people who worked on the human T cell stuff with SLE. And I want to thank all of the donors whose samples we looked at. I collaborate with a lot of people across the Penn campus and Jane Buckner at the Benaroya Institute for the lupus samples from there. And the funding. So with that, I'll stop and I will take questions. Thank you.
Thank you. That was a really clear talk. I just want to remind the audience, if you have questions, please feel free to raise your hand. We already have some questions in the Q&A. You can also type your questions there. And of course, if you would like to stay back for the coffee chat, please let us know in the chat and we'll just transition from post questions to the coffee chat on the same link. Okay, so we have two people that have raised their hands. So I'm going to unmute Bernard PR first.
Ah, Bernie!
Hi, Montserrat.
How are you, my wonderful friend?
Very nice to see you. Excellent talk, by the way.
Thank you.
I was just wondering, so you mentioned that you have this, Xist RNA binding proteins which are misregulated in lupus, and you also say that you have no Xist localization in immature B cells and T cells. So do you think it's the same mechanism? So is it the same factors which are low maybe in immature B cells and T cells, which are maybe also low in lupus, or is this maybe two different mechanisms? So
yeah, so
basically like a maturation problem of the cells.
So that's a really great question. So the way that we've been thinking about it is there could be sort of this, so in the immature, right? So as in the healthy state, as the B cells and T cells are developing and they lose Xist localization, even though it's transcribed. So I don't know if I mentioned this. So there's abundant Xist transcription during the maturation, but it's not localizing. We think of it, there could be a passive mechanism to prevent Xist, like it just can't get to the inactive X. And this could be maybe, like crowding or something, right? Or there's not enough proteins of Xist binding proteins to keep it there tethered. Or it could be an active mechanism preventing it, right? So actively like a sponge. So Xist RNA proteins that are preventing it from getting there or binding across the inactive X as to block it. We don't know yet which one of those is happening, right? And it doesn't necessarily have to be mutually exclusive depending on what the proteins are, right? There's so many proteins that have been identified thus far. And the other thing to note is that all of these different the Xist interactome thus far has been defined in differentiating mouse embryonic stem cells, MEFs, like so mouse fibroblasts, right? And I think one of the studies looked at human cells. I can't remember if it was 293. So it's a very limited type of cell, and we're thinking in the immune system, right? So there could be completely novel Xist RNA binding proteins that are at play in developing lymphocytes, in plasmacytoid dendritic cells, right, where we don't ever see Xist localized. So to kind of answer your question about the healthy state, and then in the disease state, it could be the same thing, right? So in lupus, we don't know if it's something about the way I'm thinking about it is there's a change in the architecture and the structure, the organization of the nucleus, influenced by environmental factors, that is perhaps preventing Xist from maintaining the structure of the XI territory, and that's why Xist is coming off. Or it could be an active process of something coming to get it, or the downregulation of one of these genes, like it's not there at stoichiometric amounts to keep it tethered to the inactive X.
Thank you.
Yeah.
Okay, so we'll take a few questions from the Q&A that people have written. So Mansi Apte asks, in terms of developmental trajectory, do you think DN2 and DN3 cells are trying to go back towards undifferentiated progenitors? Also, if the Xist is still getting transcribed but not localized, do you think it's getting co-transcriptionally sequestered or degraded?
Okay, so do the first question again. Let's do one at a time.
Okay,
Sorry,
yeah.
my brain is like.
No, my bad. Do you think that DN2 and DN3 cells are trying to go back towards undifferentiated progenitors?
Oh, okay, yeah, good question. So what's really crazy about this is that Xist isn't localized, but the inactive X is not becoming reactivated. So with all of these quiescent naive cells, they don't have Xist RNA localized, and they don't have a lot of these marks visual, like we won't see them at the cytological level, right? But the cells are still dosage compensated, so it still has an inactive X chromosome. But these marks using microscopy, we don't see them. Now we need to look at like at the genetic level, right? Like looking at the genes to see what's happening across the inactive X, which we're like in the middle of mapping these kinds of things. And so it's not necessarily that the DN1s or DN2s and 3s are like changing. They're different. They're continuing along a differentiation trajectory to become mature mature T cells, right? But the fact that these marks are coming on and off it's like uncoupled to pluripotency or to development, if that makes sense. And then what was the second question again? Sorry.
The second one is, if Xist is still getting transcribed but not localized in these cells, is it getting co-transcriptionally sequestered or degraded?
Yeah. So that's a great question. So we looked at the degradation. So we did half-life experiments. We did half-life calculations with actinomycin in B cells during activation. And what we found is that the half-life of Xist in B cells is like 1.9 hours. So it gets turned over really fast in these cells. In mouse embryonic stem cells and differentiating ES cells, the half-life is about 6 hours. So it's much more stable in the early development context. So it's being produced and it's getting turned over like right away. We haven't looked like earlier at the progenitors to see if it's an even faster turnover. And the other thing too to keep in mind is that there's differences with cell division, right? So during B cell maturation, there's not a lot of cell division happening. And so at first we thought, oh, well, maybe that's why Xist isn't there, because the cells they're not proliferating, like they're not dividing, right? But in the thymocyte development, there is proliferation that's happening, especially I think it's like the DN3 and the before there's active proliferation, but the marks are coming off. So localization of Xist is not tied to proliferation of the cell. Great question.
Yeah, we have a whole bunch of questions. I'm just going to say, and Carolyn Brown has a hand raised, but Carolyn, since you're joining us for coffee chat, I'm going to wait for you to be able to ask your question. Yeah, but to those still here, we're going to transition to coffee chat. If you haven't let us know yet, please tell us in the chat that you'd like to join. And if not, we'll see you next week, hopefully.